why do we do research? So it goes back to basics, really. So we need to know about the disease process. We need to understand what is happening in the disease. And the things we'd like to know about, it would be good to know, how does it start? Often we're probably never going to understand how it starts. The diseases are either autoimmune or they're complex. But we would like to know how it starts so we can pick patients who are at least early in the stage of the diseases. More like progression, what drives the disease? We can understand the drivers of the disease. We can perhaps target the disease better, work out what pushes patients down different groups. It'll be good to know what controls severity and specificity, so what dictates how bad the disease is. They're all free. In my disease, scleroderma, you get patients that have very, very progressive disease. They'll get fibrosis over a period of months, and those that last for many, many years with a very slow progressing disease. So what dictates that difference in severity from very severe, almost life-threatening disease to people that last for 20, 30 years and maintain a fairly good quality of life? And also organ involvement. Again, why different organs are involved. So in my disease, scleroderma, you get skin involvement. Some patients get lung fibrosis, lung scarring, some get kidney scarring, some get bowel scarring, some get heart scarring. We don't really know why different organs are involved in different patient groups. So it's more of a heterogeneous sort of disease spectrum, or very much like pemphigoid. And why is that? We can understand that, we can target different organs specifically. So organ-based therapies, you target the heart or the kidney or the lung and the skin separately. Or some people get all diseases, all organs involved. So it's very important to understand those key questions. That's why we do research, really. And for my own interest, I've been interested since I was at school. I did biology at school. My teacher then, I remember now, Mr. Crowhurst, it was a long time ago, Crowhurst. I just enjoyed the biology classes and working out how cells and tissues and organisms worked. I used to get good grades in biology, so I just followed that through my entire career. From my school, I went to college, got a degree in biology, went to biochemistry, so I just followed it through, partly by chance, partly because my interest was always in that area. So I want to understand how things work and try and dissect out things sort of at a cell and molecular level. If we can do that, then we can understand better the targets. I mean, John and Sarge have mentioned these different areas, the scarring, the inflammation, the B cells. Are these the best targets? Often now you want to target more than one thing. I guess one common theme which has been mentioned is if you block inflammation, you still get fibrosis. So why is that? Why, if you block what's thought to be the starting part of the disease process, that inflammatory response, does fibrosis still carry on, albeit slowly and maybe it's slightly attenuated, it still carries on. So what causes that sort of continual process? And then perhaps we can find markers for the disease. People are very keen about biomarkers now. I'm sure you might have heard this on the TV. It's a very hot area. And it's normally to do with blood samples. Can you look in the blood for a marker, probably it's a protein or a cell type, which gives you some indication that the disease is getting better or worse or changing somehow. So it's a nice, what's called sort of non-invasive biomarker that you can look at just by taking a quick blood sample and measuring a factor or a cell type in there. So that's a very sort of hot area. And they're good for two things, but they can predict the disease course. Is it getting worse? Are you getting better? Often diseases will stabilize over time. People will go into remission as a natural part of the disease, or some won't, some will progress much faster. So perhaps it can be used to sort of map out and predict disease course, and also response to therapy. Are you responding? Are you responding? Often, for example, with lung fibrosis, it's difficult to understand whether the patient's doing well. Is the lung getting better? There are tests for the lung function, but they're quite crude, blunt instruments. So we could have a biomarker to show that the lung's repairing itself better, for example. It's hard to tell often from a patient, is the lung getting better, because you can't really see it easily without doing sort of CT or X-rays, which give you doses of radiation. So that's sort of why we do research. And from my point of view, when I started as a basic scientist, I'm quite passionate about therapies, to try to get a therapy to help patients. I see patients in the corridor in our clinics, and you realise how sort of horrible these diseases are. So I'm quite keen to try and have an impact on that. Although I do understand that people like to do science for science sake and to understand diseases, understand the way that cells move and migrate in a test tube or in a culture is quite nice. I think nowadays there's much more of a drive to make it applied research. So what I do is I want to have an effect, a benefit to the community, not just because I know how a cell moves, I mean, what does that movement mean in terms of scarring or in fibrosis? So there are different types of research. I've just put sort of 3 broad areas here. So I would be classed as a laboratory scientist with a bit of translational. So I work in the lab. I'm a basic scientist. We're called basic. It's not a very. It's been used a lot. And there we look at cells, tissues, biopsies in a laboratory environment. So quite distant from the patients. It's in a very sort of unusual atmosphere when you go into the laboratory. I mean, John's a clinical scientist. He's more applied. His area would be things like epidemiology, patient stratification, doing small trials. Then there's a translational area, which is where we as scientists aspire to try and take what we do in the laboratory and then move that towards John's area into a clinical trial. So we try and understand the cells, the molecules, and the pathways that are important in the disease process, and try and pick out the key targets like the B cells and rituximab sort of thing, and say, right, that might be worth taking into a translational setting to look more closely in human samples and in patient samples, and then animal models, and maybe into a phase 1, a small trial. That's sort of a translational aspect of our work. And there's a great focus on translational research now in most research labs that work in sort of anything to do with biology or human health. And it's a bit coy, but to put what's the mission statement? Well, we want to try and convert our discoveries in the lab into patient benefit as fast as we can. Discoveries can be anything in terms of a new biomarker, a new treatment, an understanding of a disease or a disease subset. So anything that we can do in the laboratory, how can that benefit patients? So we're very keen to count this translational research program. It's been used a lot, this word translation, often overused, and people tend not to use it, but I think it does sort of sum up what we try and do. We try and translate our basic science into patient benefit, a cure, a treatment, or something that's actually useful, and rather than just being an esoteric understanding of a cell process. So that's really where we sit. And I think Jill's the same. John's the same. All translational research. So what are the approaches that we take? Well, it's all cell culture based. We look in cells, and you can take cells from tissues, from biopsies. You can grow them in the laboratory, and they all grow. They proliferate. They divide. You can grow up large numbers of cells, and you can then look at different cell types. We can look at tissue biopsies from histology. You've seen some pictures before John showed you in terms of looking at immunofluorescence. You can take these cells and tissues, and you can, if you like, we can pull them apart. You can take different cells and work out what those different cells are doing, and you can take different molecules from those tissues and ask what are those molecules doing. So an example would be, often if you're looking at a scarring process, you take out the cells called fibroblasts, and you can put them in culture, and they will start to divide and grow. They'll move around, and they'll secrete the scar tissue in the laboratory. So you can measure those parameters. You can measure how fast they move around, how fast they divide, how fast they grow, and how much they secrete, how much scar tissue they're depositing on the actual floor of the tissue culture flask. So you can look at activities of those cells which reflect what they would be doing in the body in terms of making the scarring tissue. But you're looking at that in the laboratory. And you can then, you grow these cells in a flask with what's called a culture medium. They have to be in some sort of liquid to keep them alive, otherwise they get dry and they die. They secrete things into that liquid, like the cytokines and growth factors that you may have heard of, so we can measure those in the medium as well. What are they secreting? What are the molecules that they're secreting outside, and are they secreting out more or less than a normal cell? That's sort of in vitro studies, and you can go into the preclinical models. These are often mouse models or other animal models, other animals you can use. And we really use those because when you look in a laboratory in a single cell type in a tissue culture plate, it's quite a way away from the person that their cell came from. So, for example, if we take a skin biopsy from a scleroderma patient where they have a scar on their skin, we can culture those cells in the lab, but you're quite a long way away from that cell's environment in the skin. So we try and model that by looking in animals where they have the same sort of disease process. We can have animal models for skin scarring, for example. we can look at those animal models and say, right, is the same molecule we find that's expressed at a high level in the fibroblast present in that mouse model? Therefore, is it a good model or sort of a good test of that protein in an animal model for the disease process? So we tend to use those, and we use those for testing therapies. For new drugs, often tested first of all in animal models to make sure they're safe and they don't have any side effects. And then we come to the human samples, which are absolutely crucial. One has to get back to the patient. I mean, all the in vitro studies that we do from cells, from normal cells, from patient-derived cells are all very fine, but at some point we have to get back and really ask the key question in the samples from the patients, really what's going on. Are those cells important? Are the molecules important? And without that, the rest of the work becomes, it's not really important, because if it doesn't work in patients, then really it's not going to be useful as a translation tool. So patients are absolutely vital to our research. And the more I've worked in this area, the more essential patients have become to my research. Although we still do basic science, looking at cell lines and animal models, we want to translate our work. And unless it's the same in the patient, our work just really stops. We can't go any further. So we tend to use blood and tissue biopsies. And with John, lots of tissue biopsies from the eye, lots of eye fibroblasts. And it's really a comparison. We're comparing all the time. It's sort of the main part of it. Although we do lots of different experiments, overall we're really comparing. We're comparing what's happening with disease cells compared to normal cells, or disease plasma compared to normal plasma. So we can take blood samples, we can make plasma serums, white cells, your immune cells, the B cells, the T cells that Sarge is talking about, the epithelial cells, the fibroblasts. We can take them from the tissues and we can look at them and compare them to a normal or a controlled population, or patient with a different disease. And we're doing comparisons. What's different from the normal situation? And is that difference important? Is that a disease driver that we're looking at? So it's already comparisons that we're doing. And when we count those sort of experiments, then we tend to put together what the Americans call our working hypothesis, how we think the disease starts and how it develops over a period of time, and try and just put on the slide key areas that we should be working on. Inflammation, autoimmunity, very important in pemphigoid. Tissue repair, we know we get scarring and fibrosis at the end. So these are the key, if you like, disease features, manifestations, key sort of checkpoints in the process where we think, like, we now need to work on those areas and try and block the inflammation, ultimately try and block the repair and try and block the fibrosis. And then we can just put this into a sort of understandable context for our sort of research staff to start to work on. And it's a bit abstract because patients are quite different. Some are more inflammatory than others. Some have much more fibrosis than others. But this is just a general scheme we like working to. And it's a bit busy, this slide, I'm sorry, but we look at the T and B cells there, inflammation, the fibroblasts, and the scar tissue, sort of to pick up three areas which are areas that we work on. And of course, my area is scarring and fibrosis. This is a cell that I'm interested in, fibroblasts, myofibroblast. And it's really this process, this scarring process which I'm interested in, and which is a major problem. It's one of the most severe, lethal complications of a lot of these autoimmune diseases, not just pemphigoid, but scleroderma. I work on lung fibrosis, atherosclerosis, Gill's disease, kidney fibrosis, diabetes. All have this, if you like, response where, following an injury of some sort, be it from chemical, can be a surgical injury, can be an injury due to an exposure to an environmental toxin, or an autoimmune disease, you get this repair process that takes place. And if that repair is not controlled properly, you get scarring. It's sort of, if you like, it's a common endpoint to a number of human diseases. And we can look at those fibroblasts again, comparing between a normal cell and a pemphigoid fibroblast, what they look like, how they behave, and what they make, what they express at the gene and protein level. And those comparisons give us clues to different. Indeed, the study John mentioned about the aldehyde dehydrogenase came from this sort of study comparing normal to inflamed to fibrotic pemphigoid fibroblast. That was Sarah Dale's work she did. So that's really a example of translational research program. We've gone from the bench all the way to a clinical trial, which John's trying to organise now. That's what we do on a daily basis. How has that changed in the last 10 years? Well, I feel quite lucky. I've been around at a time in science where things have really taken off technologically. I mean, we've been alive during the time where they've identified DNA. You can now do a whole genome sequencing in one day. You can sequence everyone's DNA quickly. There's been advancing technologies, so now we can do almost whatever we want to some extent, if you have enough resources, to understand how your genetics makeup works and what proteins are expressed downstream. That's a very exciting time for scientists, really. So there are lots of things that are now we can do genetics, we can genome wide, we can actually look at your entire genome and look at all your mutations, and try and work out whether they're important in the disease process. We can do multiple biomarker analysis. We can take your serum sample, we can measure 2,000 biomarkers in your serum now within 2 or 3 hours using these large platform technologies. We can look at regulatory pathways and networks using bioinformatics systems, and we can create animal models now. Because the entire genome has been sequenced of the mouse as well as the human, and we know the similarities between the mouse and human genome, we can literally make a mouse model for any disease that we want. Still technically quite challenging, but you can certainly knock out genes, any gene you want now in an animal model, and try and mimic the human disease. And of course, you're all aware with the new stem cell area, where stem cells can be used to renew tissues. And in the eye and at Moorfields here in ophthalmology, they pioneered a number of stem cell techniques to cure blindness for different blinding conditions. So very, very sort of hot topic, and one which Moorfields in ophthalmology here has actually sort of pioneered over the last few years. So it's an exciting time, I think. We can now really do almost whatever we want and try and understand the disease process, but we still need those raw materials. We need the samples from patients to actually put into these technologies. So what we can sequence genes, but we need to have the DNA from the patient to sequence from. So we need the starting materials, for that, the building blocks of our research, which are really samples from patient groups. Thank you. And sort of what's a normal lab? Well, a lab has a little bit of laboratories where people run around and work and do experiments. And labs are always, my daughters are all, Dad, you're working in research, they're all very exciting, but really when you work in this research lab, after a while, it's a lot of very routine. You get used to working in your own environment. So for us, a lab's just like an everyday place, like coming into an office to work. But if you're outside the science field, labs always seem very exciting and sort of a strange place to work. Much more work now in offices than when I was a student. I was in the lab almost all the time at weekends as well. Now students tend to sit around. We call ours the think tank. They think a lot in offices and they do things in offices. And there's much more work now in silico. You know, the internet. When I was a student, there was no internet really, and I had to go every week to the library and go through current contents, which had lists of all the journals in there. You go to each journal, read all the titles, and go to your areas and then get the journal, photocopy it, go back to your office and read them through. Now these guys sit there and in half an hour they're experts. They log onto the internet and they can get 20 papers in the field. I mean, it's too easy now. It's a different type of search, but so much more time now spent at their desk doing sort of those sort of searchings. And of course, we have rooms like this where we meet and discuss things and have nice meetings with the patients and try and get them on board. That's all the overall facilities. And then another thing which has changed when I was in my lab doing my PhD many years ago, I was working really on my own, really, with my supervisor, and we had a project up and running. We were working on it for 3 years, and that was it. Now, it's a much more team-based approach because the whole area of bioscience has exploded. So there are scientists like John, there's biologists, biochemists, geneticists, all working together now in big teams to try and answer it's big science, big data, big questions, and they all have expertise in different areas. Some people are more to do with know how cells work, some know how pathways work, some know how the genes work, some can put their data together, mathematics. So it requires collaboration now to work, both within your institute, nationally and also internationally. And each member of the team has their own expertise and skills that they bring to the table. And it's important to have all these people on board when you start to do research and to really make an impact in your area. As I mentioned, one of my roles is to be an enterprise champion for medical sciences. So trying to collaborate much more with industry through UCL and through Moorfields. And I think things have changed a little bit. I know industry can be quite challenging at times, especially when it comes to the sort of drug reimbursement area. But I guess I work slightly more earlier in drug discovery at research level before we get to that stage. Trying to get them on board, it's become easier. I mean, they realise that we are the experts now in the disease, and they have expertise in making drugs and biologics to block pathways. So I think each group brings its own expertise to the table here. And at least in my field, I think we find now it's much easier to work with the industry in more of a partnership than it was sort of 10 years ago. And so I'm trying to promote this, and they're funding PhD students, they're funding larger applications, larger grants to actually discover new drugs and new molecules to take forward into this translational program. So I'm hoping to sort of expand that over the next few years. And it benefits both sides, because industry can't work on it on their own, and they're really bad at doing research in diseases. They can make drugs and make molecules, whereas we know the disease very well, and we can tell them what the best targets are that they should then make antibodies or molecules against. So I think that sort of thing has changed. And just the last slide. It's always a headache for me. I don't work in, I have to find the funding, like John, for the group now. So our sources are government, MRC grants, but they're very difficult to get. Charities, often the grants are just as good as the grants you send to government, but you get slightly more funding because you're working in a disease area which your charity wants to promote, and more company funding now. But this is always a continual headache to try and get funding for your research. Funding is often short term. It's with a program or project grant. Shouldn't get so from 1 to 3 to 5 years. So which seems a long time, but for as John mentioned, I mean the molecule that we're looking at now, this dehydrogenase, it's taken 6 or 7 years to get together, to get from start to finish. Just take that sort of time. It takes 10 years from a research idea all the way through to a drug or molecule. So a 3-year project grant, it's still nowhere near 10 years' time. It's still a very short period of time to understand in detail what we need to understand in terms of the disease. And funding takes about 6 months to a year to get funding in place. So one has to always think ahead, funding. And although universities provide us the space and the labs, they don't give us any money to do the research. We have to get the research money from outside to bring to university. So it's always a continual challenge, and a lot of my work is spent trying to get funding from various places. And just the last slide, what is the future research? Well, I think, as I mentioned, very exciting time now. I mean, there are lots of technologies out there we can use. There are lots of companies. You can almost now, if you define a pathway in your cells, there will be a molecule out there which you can use to block it. So I think we have the opportunity, if we can understand the disease in more detail, we can then get some potential target and treatment within a fairly short period of time. And I was talking with Julie the other day about funding and the future research, and we got together the 5 Ps. We need patients, because without patients we can't really. We need a place like UCL, Moorfield, ophthalmology, all the institutes are linked to UCL user partners. They're top in the world. They're top 5 in the world. They're the same as Oxford, Cambridge, Harvard, Stanford, MIT. It's a top institute. So we need a good place. Need a project. Well, clearly working on scarring is important because we're here and we know that it's an important area. The people, it's important to have people like John and me and Jill, and also the juniors like Surya and Sarah, to actually do the research work. Some passionate research staff that actually want to go in the lab and actually understand things. And of course, it requires funding, which is always a big issue. It requires money to carry out the research. But I think in the future, it's as long as we can keep these five Ps in place, and the government are trying to ring-fence money for research, we're looking at a very exciting area, and hopefully developing some effective treatments in the future for scarring and fibrosis. Thank you.
Thank you very much.